Hello class, so this is the final part of the series of protein purification and function. So as I mentioned in the previous lecture about some protein purification procedures, so we're going to continue that discussion in this lecture. And so as a reminder, I spoke about electrophoresis within itself is not a purification method, but more of a way to analyze how pure your sample is. This gel made of polyacrylamide, then it has a pH gradient. And so when you apply potential, when that pH equals the isoelectric point, that is when the actual protein starts moving because it's at a net zero charge. But when it is charged, it actually starts to move down the gradient towards the positive end of the electrode. And so, as you also recalled, I talked about the SDS Page method, which actually uses a detergent that wraps around the protein, creating this micelle and making it electronegative, where in this aspect, it keeps the charge to mass ratio constant. And with that, when you applied your potential, the lower molecular weight travels the fastest down the gel. And you can visualize this protein by applying a dye called the Kumasi blue dye. So with this particular method, the correlation between determining the molecular weight is actually a linear type of relationship in some aspects, and therefore you are able to potentially find the molecular weight of an unknown as long as you have the molecular weight of known proteins in the solution. And being separated, you can find out what your unknown solution of protein molecular weight is. You can apply both the electrophoresis method that is isoelectric focus and the SDS method all combined in one single analysis, where once again, if you have the pH gradient and where the protein stops at its isoelectric point, and then use this as a combination with the SDS to have this two-dimensional electrophoresis, where one dimension is the decreasing molecular weight, and then another dimension would be the decreasing of the isoelectric point. Then you have the other purification methods such as salting out, where you have a solution that's full of different proteins with different solubility. The least soluble will precipitate out, and then adding some more salt will take out the second least soluble protein, and you can take that protein out after it precipitates, and so on and so forth, as trying to separate the proteins based off of solubility. You can also separate due to density of the proteins by using a centrifuge method, and where you have this stabilizing sucrose gradient, where the sample actually passes through while being centrifuged, and the protein that is most dense will be at the further most end of the centrifuge, and the least dense protein will not be as far down the centrifuge, and then from there you can do separation. And so that concludes purification methods for protein, and now we'll talk about the protein functions. Protein functions by interacting dynamically with binding to other molecules, and we can divide this interaction into two types. One type is altering the chemical configuration or composition of a bound molecule with protein that's acting as a reaction catalyst, which is known as enzymes. And the other type of interaction is totally the opposite, when other interaction is neither the chemical configuration nor the composition of the bound molecule is changed. And so this just highlights 6 of the key principles of protein functions. One is the function of many proteins involve reversible binding of other molecules. Second, the ligand binds a protein at a binding site that is complementary to the ligand size, shape, charge, and hydrophobic or hydrophilic character. Then the third one, the proteins are flexible. Fourth, the binding of a protein and a ligand is often coupled to a conformational change in the protein that makes the binding site more complementary to the ligand, permitting tighter binding. And 5, in a multi-subunit protein, a conformational change in one subunit often affects the conformation of other subunits. And finally, 6, interaction between ligands and proteins may be regulated. We'll go a little bit more in extensive details with this list as we go into the topic of enzyme, but this is just a initial introduction of key principles with protein functions. And so speaking of binding with proteins, you can have oxygen binding with protein, which is a reversible binding. And they have an example right here with myoglobin, hemoglobin, and many other proteins have a heme group. And so with this heme group, you have this porphyrin ring structure, which has 4 pyro rings. So that's right here, the 1, 2, 3, 4 with the nitrogen. That is 1 pyro ring, and then there's 4 of them. So 1, 2, 3, 4, and they are all bound by a methane bridges to hold the ring structure together to have a total of a porphyrin. And so from there, what's in the center of the porphyrin is an iron complex, and from there, that iron is what actually binds to the oxygen. And so the thing about, let's say, carbon monoxide, when there's carbon monoxide is in the air, the binding between oxygen and the carbon monoxide has a better binding with proteins versus oxygen. So oxygen is no longer being transported, and that's how you have that carbon monoxide poison, because the carbon monoxide is replacing oxygen because it has a better binding towards the carrying protein. And so thinking of binding, you can quantify this because this is basically a reaction of having proteins, having free ligands, and then now having bound ligand complex. And so this, like any other reaction, has a association constant. They describe it as Ka, like chemists doesn't have more things on their mind than think of different terminology to not confuse people. But yeah, do not have it confused with the Ka that's associated with acid dissociation constant. This right here is talking about association constant between the protein and the ligand. And so you have on the top right here association rate divided by the dissociation rate. And so with this equation, you can manipulate it where you can find the ratio of bound to free protein is directly proportional to the concentration of free ligands. And so this is the bound protein, this is the free protein, and you see that ratio is directly proportional to the concentration of free ligands. And so there is a graphical representation of the binding of ligands, as you can see right here in this chart, where you have this binding equilibrium. And so with that, you have binding sites occupied over the total binding sites. And so the binding sites occupied is the protein bound to the ligand, or the ligand bound to the protein. And then you have, as far as total binding sites, is sites that are bounded with ligands, and then protein that does not have any ligands that are bound. And so with that, you could do some mathematical manipulation that we talked about before with the association constant and free ligand, and the ratio of bound protein and free binding sites, where now you can actually times this equation both sides by the free protein, so binding sites, protein that doesn't have any ligands, and so that cancel out the P over there, and you have the P that's associated on this left side. And now you can replace the bound sites with the free proteins, and then association constant and the free ligand concentration, which is right here and right here, because you see that it directly replaces the bound ligands on to the protein. And then from there, you can take out the constant of free binding sites, so that P will come out, and that is left with this formula here. And then from there you can take out the association constant, and you're left with this equation with the free ligands over the free ligands plus 1 over the association constant. And so you can convert the association constant to the dissociation constant. And so from there you have finally the binding equilibrium. And then also just note that the dissociation equilibrium is pretty much the opposite of the association equilibrium, where now you have the free binding sites of protein and free ligands over the bound ligand on to the protein. So you have the dissociation rate over the association rate. And then also just trying to understand this binding curve. If you look at the single point, when it is lower, that means it has a lower binding affinity. And as it increases, you have a higher binding affinity. And so as far as different affinity association with different proteins to ligands, one of the examples that has high affinity is biotin and avidin. They actually utilize this interaction when trying to create new biosensors, where they have something that is templated that should work, and then from there they start replacing those interactions with different DNA, but as long as they see the sensor within its fundamental template actually works with something that is well known, then they go on to the next stage of doing interactions that are not well known. And when it doesn't work, we know that it's not the device itself, because they tested it with the standard biotin and avidin type of interaction. And one final thing is the effects of pH on to oxygen binding to protein. When you have a lower pH, you'll see that there is a lower affinity, and a higher pH is a higher affinity. Not to say that this is for all cases, but this is just a general understanding of there is effects to the binding of oxygen to protein affected by pH. And that concludes the protein purification and the function lecture.